Okay, good evening, everyone. Uh, so we'll start the class now. Meanwhile, others can join the call. Okay. So today is the last session for this course, Experimental Biotechnology. So this is an extra session that I'm taking apart from the 12 uh, week sessions that have already been uploaded. Uh, so today basically is a revision class plus uh, you can say like you can whatever doubts you have in any of the topic we can discuss in today's class. So today's class will be only for 1 hour, and if you have any form of doubt in any of the sessions previously discussed and any of the questions that have been previously discussed, we can discuss it today. I'll try to clarify your doubt. Okay. So today also I have prepared some questions which are basically related to the questions that have been discussed previously. So I'll be discussing those questions as well as I'll give you time to ask any question if you want. Okay. So let me begin with asking you all if you have any doubt or any particular topic to you want to discuss. Uh, okay, Aditi has asked CBB and what is CBB, Aditi? Can you clarify? Okay. Oh, sorry. Kumasi and colloidal Kumasi compatible mass spectrometry. No, actually, when you have to do mass spec, you have to destain the gel completely. I'm assuming you are asking like after running the gel, if we slice up the gel and then analyze the protein into mass spec, right? Yeah, so in those cases, generally what we do, we destain, we remove the staining components completely because they kind of interfere with the mass spec results. So if we have done Komasi staining or if we have done uh, silver staining also, there are procedures to remove those stains in order to do the mass spec analysis. Generally, when we are going to do mass spec analysis, before that we prefer doing silver staining because it is more sensitive. And after that, there are procedures to remove the silver stain. So the gel becomes, so you can do the silver staining. You have the stained band. You can excise it, and then that cut gel part that you can do destaining. It would be a transparent gel uh, piece, and then you can homogenize it and inject it into the mass spec. Okay. Anyone else wants to ask any question, or should I start with the questions that I've prepared? Okay. Protein is treated with 2 molar and 8 molar urea. The retention time of the different species in increasing order would be dash. Uh, so basically, as you increase the uh, urea concentration, uh, so as you increase the urea concentration, your protein will keep unfolding, right? So they will uh, the retention time of different species. The native partially unfolded and native will have the highest retention time. Yes, yeah, the correct answer should be C. So the unfolded, wait, no. So basically, the folded protein, the completely folded protein will elute last. from the column because it will be the smallest molecule. The larger molecule will elute first from the size. Like I'm assuming this is talking about size exclusion column. So the largest molecule, that would be the completely unfolded protein, that will elute first, then the partially unfolded, and then the native protein. Yeah, so C will be
Hi,
the right.
ma'am.
Yeah,
Uh, yeah. So the unfolded one will have the lowest retention time,
yes,
right?
yes, 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 yeah.
So I think C will be the
C
correct
will be the
answer.
correct answer, yes.
Yeah, actually in the solution it was given A, so I got confused.
This question is from
This is
my set only.
no, this is one of the question from assignment 5. Last question from assignment 5.
The assignment that is given on the portal, or the
Yeah,
assignment that
right.
I have discussed,
No. The assignment which is
that is
given in the
given on the
portal.
portal. Okay,
Right. So there the correct answer was given as A. So I got confused because I have ticked C.
okay. And it has, like, I have seen some of the questions in some of the assignments have been reanalyzed, and like mail has been circulated, like for these questions the marks was given to everyone. So this was not the case in for this question.
No, I don't think so because this is the work solution that they have given last. The work
Okay,
solution was given a few days back.
okay,
So there it was given as C. So sorry, A. So I got confused.
okay.
So I just clarified. Anyways,
Yeah,
that will be the C.
yes, I think it should be C. Basically, the smallest molecule will elute last, so it will be having the highest retention time. Yes,
Right. Thank you, ma'am. That was
yeah.
my logic as well. Okay. Thank
Huh.
you.
Okay. Anybody else wants to ask anything? Okay, I'll start with the questions that I have prepared, and meanwhile, if you have any other doubt also, you can write in the chat box. I'll try to clear them up, okay? So starting with the first question today. How many grams of sodium dichromate should be added to 50 ml water to prepare 0.025 molar sodium dichromate solution? And the molecular weight for sodium dichromate is also given. So I have already discussed such type of questions in the class. So I request all of you to try to calculate this using your calculators, and let's see if you can solve this. So today I'll be discussing the questions that are kind of related to all the sessions. So this will be kind of a practice for your exam as well. Okay. So requesting everyone to try to solve this. I'm giving you 2 minutes. Like just give me an answer, whatever you are getting. Okay, we have two answers in the class. Aditi is saying A and Rania is saying B. Aditi, did you calculate it for 1 liter or 50 ml? Okay, it's okay. I think you understood your mistake. So, correct answer is option B. So, I'll show you how to calculate this. So, basically, the molecular weight is given as 261.97 grams per mole. That means 1 mole has these many grams of sodium dichromate. Okay. So, so if I have to prepare 1 molar solution of sodium dichromate, that means it will contain these many grams of sodium dichromate in 1 liter of water, because 1 molar is basically equal to 1 mole per liter. Okay. So, the molecular weight is 261 grams per mole. So, 1 mole has 261 grams. Okay. Now, 1 molar means 1 mole per liter. Like in 1 liter, 1 mole of the molecule will be present. So, 1 mole of this compound is 261.97 grams in 1 liter of water. So, that would be 1 molar. Okay. So, now from that we can easily calculate 0.025 molar. So, 0.025 molar would be this into this, and this calculation that we are getting is for 1 liter of water. Now, pay attention to the question. In the question, I have asked how much do we have to add in 50 ml of water. So, in 1 liter, you have to add 6.54. So, for 50 ml, you can calculate. So, 1 liter means 1000 ml, right? 1 liter is equal to 1000 ml. So, for 1000 ml, you are adding this much. So, for 1 ml, you can calculate like that, right? 1000 ml, in 1000 ml, we are adding 6.54 grams. So, in 1 ml, we will be adding 6.54 divided by 1000 grams. So, in 50 ml, we can add 6.54 divided by 1000 into 50. So, that is what I have done. So, 0.327 would be the correct answer. So, 0.33 approximately. Okay. So, that is how you solve such kind of questions. So, I have given such kind of question in the first week session also, as well as in the last session that I have taken on Friday. So, I'm emphasizing on these questions because these are very basic kind of question, like not just for this exam, but in your life, whenever you are going to do biotechnology experiment, this is something that has to be used by you irrespective of whatever experiments you are doing. You will have to make some kind of solution or some kind of buffer. So, these questions should be very clear to all of you. Okay. So, anybody has any doubt in these kind of questions, you can still ask me. So, today is the last session, so ask me now, otherwise it will be difficult for us to clarify any doubts. Okay. Clear to everyone? Okay. I'll move on to the next question then. If anyone has doubt, you can ask. The correct answer is option B for this one. Okay, moving to the next question, question number 2. In SDS page, protein sample is first treated with detergent sodium dodecyl sulfate in order to, option A, make the protein become negatively charged, option B, make the protein become positively charged, option C, renature the protein, and option D, adjust the pH of the protein. Priyadarshini, do you have any question? You are raising your hand. Uh, Priyadarshini. Do you want to ask anything? You have raised your hand. Okay. Sure. So, all of you have answered option A. Most of you have answered option A for this question. So, I think this concept is pretty clear to you why we are adding SDS and SDS space. So, SDS is the molecule that gives negatively charged, that gives negative charge to the protein, as well as it denatures the protein. It basically unfolds the protein into a single amino acid chain so that it can be resolved in the polyacrylamide gel based on its size. Okay. In nature, proteins are completely folded, so they have a smaller hydrodynamic radius as compared to its unfolded form. So, unfolded form can give us the exact molecular weight of the uh, protein. Okay. So, all of you have answered option A, that is the correct answer. So, this was the SDS structure. Sorry, this is the SDS structure. So, here we have the hydrophobic tail and the hydrophilic head. The head has the negative charge on it. So, hydrophobic tail interacts with the hydrophobic inner parts of the protein, unfolds it, and then the hydrophilic head that is having the negative charge imparts a complete negative charge on the protein molecule. Okay. Moving on to the next question now. Question number 3. A researcher is working with a protein that contains 4 subunits of differing molecular weight. If the researcher performs native page and SDS page, how many distinct bands should he see on the gel? So, respectively, you have to answer how many bands on native page, then how many bands on SDS page. Option A says 1, 4, option B 1, 2, option C 4, 1, and option D 4, 4. Aditi has asked a question, ma'am. Is this the only difference between urea and SDS page imparting? Uh, Not exactly. So, urea page can also be done in presence of SDS. So, urea page basically is done to understand the folding nature of the protein. So, urea denatures the protein completely, and as you increase the urea concentration, you can see how quickly a molecule is unfolding, or like if it is in a partially folded state. Okay. Whereas SDS is something that SDS is an anionic detergent. Okay. Unlike urea is not a detergent. SDS is an anionic detergent. That's why it has both hydrophobic and the hydrophilic uh, parts to it. So, that detergent is added to the protein in order to run the gel, in order to separate the proteins just completely based on size. So, both are kind of different techniques. Okay. So, don't confuse one with the other, like both of them have their own purpose. Uh, okay. Okay. For this question, 4 people have answered option A. Others have not answered. Anyone else wants to answer? Okay, so option A is the correct answer. So you are right. So in a native, the difference between native and SDS pages, presence and absence of denaturating and reducing agents. So in SDS page, we have SDS and beta mercaptoethanol or DTT in the loading die in order to completely reduce the disulfide bonds and to denature the proteins. Whereas in native page, we don't add those things. So the separation is based on both charge and size. In SDS page, the separation is solely based on size. So if the protein has 4 subunits, since we are adding beta mercaptophenol to it, the 4 subunits will be separated because all the disulfide bonds will be broken. Whereas in native page, everything will be in one single unit and you will see one single band for the protein. Okay, so one band for native page, 4 different bands because 4 subunits have different molecular weight. So 4 different bands for SDS page. Okay. Moving on to the next question now. A student researcher overexpresses an exogenous protein in cell culture and wants to determine if that protein is in fact overexpressed. What technique would best demonstrate that this protein is expressed in these cells? Okay, most of you are answering option B, that is Western blot. So you guys are correct about that. So where do we use Western blot for? We use Western blot for detecting protein expression. Where do we use Northern blot for? Why, like, for what molecule can we use Northern blot? Anybody can answer. For which molecule can we use Northern blot? Okay, RNA. And for we can use Southern blot for DNA. Okay. So Western blot is the correct answer because Western blot is a technique that is used to check the protein expression. So just to give a quick revision for Western blot, we have, let's say, a cell lysate expressing a particular protein that we want to detect. We can separate the protein on SDS page first. So our protein of interest will have a specific band. Now we want to see that protein, like we will see multiple bands when we are doing SDS page, but we want to particularly identify our protein of interest. So we'll have the antibodies for our protein of interest. We'll bind the antibodies to that. First we'll blot the gel on a membrane and then we'll bind the antibodies onto the membrane. Wherever our protein of interest will be present, the antibodies will go and bind. Then we'll uh, detect that antibody by using a secondary antibody that will give some kind of color, and then we can see that whether our protein of interest is expressed or not. Okay, so that is the Western blot technique. Anybody has any kind of doubt in this technique? So now northern and southern blotting is also done in the same way, just the molecule, instead of running a SDS page gel, we are running an agarose gel for DNA or RNA, whatever the molecules you are trying to separate. For RNA, we generally use denaturating agarose gel. So you can go through the lectures to understand the technique in detail, but the principle kind of remains same. For detecting DNA and RNA, for in Western blot for detecting proteins, we are using antibodies. In northern and southern blot for detecting DNA and RNA, we are using different oligonucleotide probes. Okay. Anybody has any doubt in these techniques? In our exam, can we have questions like explain the detailed protocol? I'm not sure about the exam pattern. Like are just asked to take the doubt clearing session. So the NPTEL team will be able to clarify these doubts. If you have any kind of forum where you can ask these questions, you can ask. But I'm not sure, or it should, like, as far as I'm guessing, it should be MCQ, but like either Google it or something. I really don't know what is the exam pattern of this. Okay, sir told you that it's MCQ, then it should be MCQ. Okay. Anyone else has any kind of doubt in these techniques? So this is just what I explained. Okay, next question. Cell line was engineered to contain and express a DNA sequence frequently associated with pancreatic cancer, with the hope that the cell line will undergo rapid cell division much like cancerous cells. You want to test whether or not the cell line is efficiently transcribing this DNA by measuring the quantity of mRNA produced. Which of the following method would be best suited for this testing this? We have just discussed this, so all of you should be able to answer. Which of the following method would be best suited for testing mRNA concentration? Option A, northern blotting. Option B, western blotting. Option C, thin layer chromatography. Or option D, southern blotting. Yes, we have two people answering option A. That is the correct answer. So just now I explained, northern blot is for RNA, western blot is for protein, southern blot is for DNA. Okay, so option A is the correct answer. Which one of the following is the basic principle of Sanger's DNA sequencing method? Option A, chain termination by incorporation of dideoxynucleotides. Option B, chain elongation by incorporation of dideoxynucleotides. Option C, release of inorganic pyrophosphate. Option D, chain cleavage by modification of dideoxynucleotides. Which one of the following is the basic principle of Sanger's DNA sequencing method? Come on, others also answer. I got only two answers. Which one of the following is the basic principle of Sanger's DNA sequencing method? Chain termination by incorporation of dideoxynucleotides, chain elongation by incorporation of dideoxynucleotides, release of inorganic pyrophosphate, or chain cleavage by modification of dideoxynucleotides. Okay, so we have three answers saying option A, chain termination by incorporation of dideoxynucleotides, which is the correct answer. Those who haven't answered, do you have any doubt? So basically, dideoxynucleotide means the 3' OH is all, like, the oxygen of the 3' carbon is also lost. So we have generally the nucleotide that is incorporated in the DNA is deoxynucleotide that has a 3' OH group. But in dideoxynucleotide, we have the 3' OH group also removed from this position. So from this 3' OH, the next phosphate group reacts, and then we have the phosphodiester linkage like this. So we have the OH and then the then the phosphate group of the next nucleotide, that both of them react to form a phosphodiester linkage. But here, due to the absence of OH group, the chain termination happens. Basically, when the primers are added and the elongation is happening, if the instead of DNTP, if the DDNTP gets incorporated, the chain will terminate at that position. And based on multiple fragments that we will be getting, we can actually determine the sequence of the DNA. So we can have 4 reaction vials with each having all 4 DNTPs along with an additional DDNTP. So we can have a reaction for A, C, T and G, and then we can run gel for all 4 reaction vials, and then we can analyze the sequence based on the fragments that we are getting. So the smallest fragment that we will get will be the first base, then the next will be the second base, and so on and so forth. Okay. Those who haven't answered, do you guys have any doubt in this, or I hope that's clear to everyone? Okay, great then. Moving on to the next question now. A DNTP master mix is prepared by combining 40 microliter of each 20 millimolar DNTP stock consisting of all 4 nucleotides. 4 microliter of this DNTP master mix is added to a PCR mix, and the final volume is adjusted to 50 microliter. The concentration in micromolar of the total DNTPs in the PCR mix is. So again, I will give you 2 minutes and request all of you to calculate this. Use your notebook, pen, calculators and try to calculate this. Just pay attention that the answer has to be given in micromolar, okay? So whatever you calculate, convert it into that unit as well. So you have the DNTP master mix that is prepared by combining 40 microliters of each DNTP. Each DNTP has 20 millimolar concentration, the stock concentration. Then from the mixture, we are taking 4 microliter and adding it to 50 microliter. Okay. So tell me the final concentration of the DNTP. Everybody try to calculate. You can type whatever answer you are getting, even if you are not getting anything from the options. Let me just see what's your thought process. If you are doing the process right and if there's any calculation mistake, that can be rectified. But if you're like whether you are understanding the concept or not is important. Could anyone solve? Okay, we have one answer from Rania saying option D. Okay. Okay, Aditi says 160. Anyone else? Okay, I'll tell you how to solve this. The answer is 1600. So, Rania, you are correct. I'll tell you how to calculate this. So, basically, the master mix is prepared by combining 40 microliter of each 20 millimolar DNTP stock. Means we have 4 stocks: DATP, DCTP, DGTP, and DTTP. All 4 of them, the concentration is 20 millimolar. Okay. So, if I am taking 40 microliter of each, that means 40 microliter of this, 40 microliter of this, 40 microliter of this, and 40 microliter of this. So, this gives me a total volume of 40 plus 40 plus 40 plus 40. That means the total volume is 160 microliter, right? Now, concentration of each uh, DNTP. So, concentration of DATP in total 160 microliter would be 40 microliters into 20 millimolar. This is basically M1V1 into M2V2. Okay, M1V1 is equal to M2V2. So, 20 millimolar into 40 microliter is equal to we don't know M2, we have to find out the M2, into V2 is 160 microliter. So, if you solve this, this will give you 40 into 20 divided by 160, right? So, basically M2 will be equal to 5 millimolar, right? This is clear to everyone. Now, if M2 is 5 millimolar and we have 4 of these, so this will also be 5 millimolar, this will also be 5 millimolar, and this will also be 5 millimolar. That means total DNTP concentration is 5 into 4, because we have 4 DNTPs. So, we will, the total DNTP concentration will be 20 millimolar. Now, the stock concentration, the DNTP master mix concentration is 20 millimolar. And we are taking 4 microliter of that master mix. That means we are taking 4 microliter of this 20 millimolar. Again, we will have M1V1 is equal to M2V2. How? We have 20 millimolar master mix multiplied by 4 microliter that we are taking, and the fine M2 is what we have to calculate, and the final volume that we have, V2, is 50 microliter. So, now 20 into 4 divided by 50. How much will it be? It would be 1.6 millimolar. So, M2 will be 1.6 millimolar, and 1.6 millimolar is basically equal to 1.6 millimolar is basically equal to 1600 micromolar. So, in the question I've asked, what is the concentration in micromolar? So, from millimolar, so 1 millimolar is equal to 1000 micromolar. Okay. So, this is clear to everyone. Aditi, do you understand now? Okay. So, the correct answer is option D. Moving on to the next question now. Question number 8. Match the stationary phase column 1 with its corresponding chromatography techniques that are given in column 2. So in column 1 we have protein A, protein A, Cefidex, phenylcephalose and diethyl aminoethyl cellulose. And in column 2 we have size exclusion chromatography, ion exchange chromatography, affinity chromatography and hydrophobic interaction chromatography. So you have to match the column 1 to column 2. Okay, so I have many people answering option B. So let's see. Option B says P3, that means protein A is affinity chromatography. Q1, so Cefidex is size exclusion chromatography. R4, phenylcephalose is hydrophobic interaction, and then diethyl aminoethyl is ion exchange chromatography. Yes, that is the correct answer. So option B is the correct answer here. So we have protein A that is used as affinity chromatography matrix for purifying. Anybody knows why? Where do we like what molecule can we purify using protein A? Yes, Rania has answered Ig. Yes, and basically immunoglobulins, antibodies are immunoglobulins. So all immunoglobulins have this affinity towards a specific protein called protein A. Uh, it is isolated from a bacteria. I forgot exactly from which strain. So protein A is used in the matrix, column matrix, for specifically purifying immunoglobulins or antibodies, as we said. Okay. Then comes the Cefidex. Cefidex is a size exclusion chromatography. It is basically dextrin polymer, and it has different pore sizes. So based on the pore size, you can have purification of different kinds of, like different size range of molecules. Okay. Then comes the phenylcephalose. I think so, Staph aureus. Yes. Then comes the phenylcephalose. Phenylcephalose are used in hydrophobic interaction chromatography, where they have the hydrophobic aromatic group that can interact with the hydrophobic molecule, bind them, and then later you can release the binding by decreasing the hydrophobicity, okay, of the solvent. Then comes the diethyl aminoethyl cellulose, which is basically a ion exchange cellulose. Anybody can answer is whether it is an anion exchange or cation exchange resin. Diethyl aminoethyl is used for anion exchange or a cation exchange. Yes, it is used for anion exchange. That is correct. That means it has a positive charge. Okay. So it has the amino group in the quaternary state. So it has a plus charge on the nitrogen, which makes it a suitable candidate for using it in anion exchange chromatography. Okay. So most of you answered this correctly. So I'm assuming this is pretty clear to everybody. Moving on to the next question. To what power would a microscope magnify an object if the 10x low power objective were in place and the microscope has a standard eyepiece? What power would a microscope magnify an object if the 10x low power objective were in place and microscope has a standard eyepiece? Okay, Mansika has answered option D. Other people also answer, then we'll discuss. Okay, three people have now answered option D. Uh, can any of you write the magnification that we get from a standard eyepiece or the logic behind 100x? Low power is 10x, that's standard eyepiece. Yes, so standard eyepiece is of 10x. So basically, yes, you guys are right. So basically, we have a 10x objective and then the eyepiece is also 10x. So the magnification is multiplicative always. So we have 10x multiplied by 10x. So that's why we get a 100x of magnification. Okay. So we have the eyepiece here that gives a 10x magnification and the objective is here over below the specimen in case of a standard light microscope. So we have 10x from this, 10x from this. So 10. So this is multiplicative. So that's why we get 100x magnification. So option D is the correct answer. Moving to the next question, question number 10. Which of the following is used to focus the electrons when using an electron microscope? Which of the following is used to focus the electrons when using an electron microscope? Option A, lens. Option B, reticle. Option C, magnetic field. And option D, phase plate. Okay. Most of you are answering option C, that is magnetic field. So, yes, you are right. So, in an electron microscope, uh, electrons are focused using magnetic field. So, in a light microscope, we focus the light using a lens, right? So, similarly, in case of electron microscope, we need something to focus the electron beams on the specimen. So, for that, we use magnetic field. The charged electrons uh, can be focused in under the magnetic field. So, these are the large magnets that are attached, like that are present below the electron gun. So, this is the diagram for a scanning electron microscopy, and this is a diagram for transmission electron microscopy. So you have the electron source where the electron beams are focused towards the specimen that is kept here using these uh, magnets, and then the electrons are then reflected back. Secondary electrons are emitted. Secondary electrons are then captured by the detector, and then later we get the image photograph of the sample. Yes, so electromagnets also create a magnetic field at the end. So the reason why the electrons are focused, because electrons have their own charge, and under the magnetic field those charged molecules can be focused into one specific direction, like a beam, like a ray of light. Like in case of light microscope, you have the lens, the light hits and the light gets condensed onto one focus point where you can keep your specimen, and then later you can see the image of that specimen. So similarly, for focusing the electrons, we use magnetic field. Okay, so this is the diagram for scanning electron microscope, and here we have the transmission electron microscope. The difference between both of them are, in scanning electron microscopes we have the electrons deflecting back from the specimen, whereas in transmission the electron beams transmit the specimen and reaches the detector that is present below. Okay, moving on to the next question. Which of the following is Edman reagent? Which of the following is an Edman reagent? Option A, phenyl isothiocyanate. Option B, CF3COOH. Option C, FDNB. And option D, phenyl thiocarbonyl. Which of the following is Edman reagent? So we have three answers, and all of them are option A, that is phenyl isothiocyanate. That is the correct answer. So Edman reagent is used for where do we use Edman reagent? Anybody? Yes, sequencing amino acids. So basically, for protein sequencing, we use Edman reagent, that is phenyl isothiocyanate. So option A is the correct answer. Moving to the next question. Edman degradation is used for, so you already answered protein sequencing. So where do we use? Option A, identifying N-terminal amino acids. Option B, identifying C-terminal amino acids. Option C, identifying amino acids. And option D, identifying carbohydrates. Edman degradation is used for dash. Whoever knows the answer, please write. We have 4 answers already, all of them saying option A. Okay, 5 now. Everybody, whoever knows the answer, type it. So all of you are answering option A. So that is the correct answer. It is used for identifying N-terminal amino acids. So Basically, this is phenyl isothiocyanate that I've discussed in the previous question. It basically reacts to the N-terminal amino acid of whichever peptide forms a cyclic compound, and then that compound can be cleaved off from the rest of the peptide. Take new N-terminal and now this will have different R groups. So phenyl thiohydrantoin, this will have different R group for different amino acids. Okay, with glycine, there will be an hydrogen in place of R. If there is an alanine, there will be a methyl group CH3. So different R groups will form a different phenyl thiohydrantoin derivative, right? Now, based on that, if we run a standard first for different amino acids, then we can compare, okay, what is the N-terminal amino acid for this peptide, and we can reproduce the sequence of the whole peptide by detecting. So we can detect the first N-terminal amino acid, cleave it off, rest of the peptide we will get, then again the next amino acid, so that will become the N-terminal now. So on, you can move on to the last amino acid and detect each amino acid by the sequencing technique. Okay. So this was the last question that I have in this session. So anybody has any doubt? Rania has written, can we have any question which has more than one correct answer? Uh, like I'm assuming you're asking for exam, right? So if there is such a question, it will be mentioned there. Like this question can have more than one correct answer, like more than one options can be selected in such question. So that option will be there. If you can select more than one options, then there is a chance that it's a multiple answer question. Otherwise, it's a single correct answer question only. Okay. So that thing will be mentioned. Anybody else? So this was the last question. So if you guys have any doubt, you can ask me now. And in any of these questions or in any of the questions that have been previously discussed in all the 12 sessions that I've taken, or any particular topic you want me to discuss, I can have a short discussion on that. Any doubt in any of the techniques? So we started with electrophoresis, we moved on to chromatography, then we moved on to immunoassays, different immunoassays. So today I have not discussed ELISA, but then we have studied about ELISA also, and then we have studied immunodiffusion, ELISA, antibody generation. Then we have discussed about cell biology techniques, molecular biology techniques, DNA sequencing platforms. So any doubt in any of the techniques? Aditi is asking stacking mechanism of page briefly. So yes, that I can explain. Let me open a blank slide. I can write here. So stacking mechanism basically is required because we have a vertical gel. Okay, the reason that we need a stacking mechanism is because we have a vertical gel. We have a vertical gel, we are loading the samples in a vertical manner. So let's say my sample has 4 different components of 4 different sizes: A, B, C, and D. And I want to compare what are the sizes. Like I want to know if A is smaller than B or larger than B, C, D, what is the size of different molecules A, B, C, and D. Now, if I'm loading it in a vertical gel, uh, there is a chance that they are not present all in the same line, but they are present like A, B, then C, okay, and maybe D is on top of A, something like that. So they are not on the same line. Now, let's say if I ask you to run in a race, you and another contestant, let's say. So, and I ask you to start from different starting points. So, will it be fair for both of you to start from different starting points and then reach a same finishing line? So, here in this case, the finishing line is this. I want to see whether which one is running faster, which one is running slower. Okay. So, if I don't start them at a single point, at a straight line, it would be difficult for us to compare which is actually running faster, like which is the smallest molecule that is running faster, which is the largest molecule that is running slower. For that purpose, we use the stacking gel. Now, what we do is we make the stacking gel of pH 6.8, and the buffer that we are using has the pH of 8.3 that has tris and glycine. Okay. So now tris molecules are basically tris plus chloride ion, and glycine has a glycine gets differently charged at pH 6.8. So 6.8 is a lower pH. Okay. So at that pH, glycine does not have a negative charge so that it can move faster. So what happens? Glycine moves very slowly, and the chloride ions being negatively charged, they move very fast. So the proteins that are loaded in this well, they kind of get sandwiched between the chloride ion and the glycine molecule. Okay. The proteins that are there, they get sandwiched between the chloride ions and the glycine molecule. So the protein gets stacked between the chloride ion and the glycine molecule, and they come in a straight line. Then from this point onwards, the resolving gel starts, and the resolving gel has a pH of 8.8, where the glycine also gets a negative charge. So all of them move very fast, and the proteins are moving solely based on their charge. Sorry, size. Okay. That's clear to you, Aditi? Do we have stacking gel in the native page as well? Yes, we have stacking gel. We have to have stacking gel in any kind of vertical gel electrophoresis. Otherwise, we cannot compare the sizes. If even if we are doing native page, if we have two different samples and I want to see the molecular weight of the native protein, okay, not the denatured protein, but the native protein, I want to see the molecular weight of the conformation, like which has a smaller conformation and which has a larger conformation. The one that is having a smaller conformation will run faster than the larger one, right? So if I don't start them from a same point, it won't be a fair competition. So this is the starting point, this is the ending point. Okay. So if I don't start them from the same starting point, I don't know if they are running at different rates because I did not start them from the different points, or they are running at different rates because they are different in size. So for that, we need stacking gel in any of the vertical gel electrophoresis. Okay. The mechanism of stacking could be different. There are other buffers also that are used apart from trisglycine. So, but the basic principle remains the same. The protein gets tagged in a single line. The molecules could be different in the buffer components, but trisglycine is the most common thing that is used, and it is something that is very commonly available in all the labs. Okay. You said two samples, so that is that two different proteins. Yes, two different proteins. If I want to compare, let's say I have two different organisms uh, growing under the same condition, and I want to see the difference between a particular protein, like whether this organism A and organism B, both of them have a particular protein that I want to compare. Okay. Now the protein from organism A and the protein of organism B has been isolated. Now I'm running a native page to compare whether the protein of organism A is larger than the one that organism B has, or it is of the same size. Something like, if some experiment has to be designed in such a way, then I have to run a native gel to determine the sizes, right? So same kind of experiments can be done on size exclusion chromatography also. I can determine the molecular weight from that also. But I'm just giving an example. Like if I want to compare two things on the native gel, I have to start it from a same starting point. Is that clear? Anyone else has any kind of question? Any of the techniques? If we don't have any doubt, then I think we can end this session. Ma'am, since this is your last day, is there any means by which we can stay connected? Uh, you can mail me on my email ID. I'll type my email ID here. So you can just mail me maybe if you have any kind of doubt. So I'm a student at IIT Kanpur. Okay. Thank you so much for joining. I think